good zombie movie. I also love a good zombie movie. And I think the fascination and horror of this, just this obsession with, oh, what if there's some type of disease or virus or fungus or something that can take over our brains, something that makes us relinquish our autonomy. I think that the reason that zombie movies are so popular is because they play on this natural fear. And any good thriller has to be at least a little bit realistic. So, where's the truth in this? So, my first point is with fungus. How many of you guys have a sugar tooth? No? Okay, well, Candida yeast is, it manifests physically and also neurologically in humans. And some of the side effects, if you have Candida in your body, can be leaky gut syndrome, autoimmune issues, but it can also cause sugar cravings. So, in a way, it is controlling your mind a little bit. But I'd say that it's rather minuscule in the grand scheme of things. Like, if you're trying to go on a diet, that kind of sucks. But other than that, it's not like you're gonna eat your lab partner. So, so when I think of more zombie-like, to me, would be parasites. And what comes to mind is the hairworm parasite, which infects crickets. Once it's ingested, it slowly eats them from the inside out and then makes them commit suicide by jumping into water so that it can lay its eggs. And this plays into the parasite manipulation hypothesis, which is exactly what it sounds like, that parasites manipulate their hosts in order to further their life cycle and further transmission to accomplish a goal. And the effects that parasites have on their hosts can be direct or indirect. And going with the hypothesis that they are purposefully manipulating you would fall under direct mechanisms, which affect neurological systems or muscles, and the parasites purposefully have some sort of impact on their host, get them to do some behavior so that they can survive. They also employ indirect mechanisms sometimes, and this is more just a byproduct of your immune system trying to combat these parasites. And it's more of a domino effect of something that your immune system has done, resulting in a certain host behavior. And it doesn't really benefit the parasite at all. So during our intellectual journey, trying to find maybe something that would affect humans the way that parasites can affect crickets, Jackson and we kept hearing about Toxoplasma gondii. And this is a single-celled parasite. Its definitive hosts are Felidae, your cats, and that's where they reproduce. But intermediate hosts can also be most mammals and birds, which includes you. And actually, an estimated 1/3 of the world's population is infected by this parasite. So why are we not freaking out? Probably because most of the people who have it are asymptomatic. And those who do have symptoms, it usually just feels like the flu. In rare cases, if you're immunocompromised, it can have worse effects. But for the most part, what happens when someone with a normal immune system has this parasite is, in order to combat it, the immune system turns these active parasites into inactive parasites in the form of microscopic cysts, and these stay in the tissues of your body, including your brain. And we refer to these inactive parasitic cysts. The condition is latent toxoplasmosis. So another intermediate host of this parasite is rats. And the effect that this parasite has on rats is that it makes them attracted to cat pee, which I'm sure you're thinking, ew, first of all. And second of all, how and why does this happen? So, the parasite T. gondii directly interacts with the rat's genes that control the production of vasopressin. Vasopressin is located in the olfactory bulb cells, which then connect to cells in the amygdala. And the amygdala is the control center for fear and aggression, so this increased production makes the rats less fearful. Now, in a normal rat, they are naturally afraid of the smell of cat pee. They will run in the other direction every time, even if they have never come into contact with a cat in their life. But in the infected, they're attracted to it, which makes them more likely to come into contact with a cat, and therefore more likely to be eaten by their natural predator, so that the parasite can be ingested and is able to reproduce in the cat. So this leads us to the question, we know that T. gondii is affecting rats. Is it affecting our minds? So there have been studies that have linked toxopositivity to a lot of things, which include slower reaction time and more car accidents, decreased fear in risky situations, increased aggression in men, women displaying more trusting and forgiving behavior, greater suicide rates in women, and schizophrenic people have a greater proportion of toxopositive people than the general population. In other words, if you were to take a group of schizophrenic people, there's more likely a chance that more of them have toxopositive people than the same number of non-schizophrenic people. Of course, correlation does not equal causation, and it's difficult to determine causation because it's just not entirely ethical to test humans the way that we've tested rats for these phenomena. But we have to wonder, how are these variables related? Does one cause the other? Or is there a third unrelated variable that affects both of the conditions? So I'm going to hand the metaphorical microphone over to Jackson, and he can tell you a little more in depth about the T. gondii life cycle and biochemistry.
All right, let's get into it. Firstly, I'd like to discuss this weird word here, gondii. Gondii is a Latinized version of an Arabic word of gundi, which is a type of rat that this toxoplasma was first found in. It has no good pronunciation. There's at least 3 valid pronunciations from gondii to gondii to gondii. For simplicity, I'm just going to call this T. gondii for the rest of the presentation. Now, T. gondii is a single-celled eukaryote, and it is an obligate protozoan parasite. Now, what that means is, one, it's a protozoa. It falls under the same general type of animal as amoebas do. They're big, one-cellular animals, and they're obligate parasites, so their entire life cycle is completely dependent on them infecting other animals. Their individual cells are quite small, only 2 micrometers by 6 micrometers wide, and they can form cysts in their hosts that are from anywhere from 5 to 50 micrometers wide. Now, for context, the average human cell is anywhere from 10 to 100 micrometers. Now, what this means is that a lot of T. gondii can fit inside a single human cell, and also they are small enough and tricky enough to actually pass the blood-brain barrier in humans and other animals, which is a very unique characteristic. Most animals cannot do this. Now, the life cycle of T. gondii is, in my opinion, needlessly convoluted. They reproduce sexually in cats, as Jamie said, and asexually in most mammals and birds. Cats become infected when they eat their infected intermediate hosts, like rats and birds most commonly. And they infect the feline intestinal cells, reproduce in them, and then create oocysts, which are just basically big eggs that pass through the cat's feces, infecting water and rats and birds along the way. Once infected, T. gondii develop into cysts in the muscle and brain tissue of its intermediate host. Now, human infection. Humans can become infected most often by eating undercooked, infected meat or by being exposed to unwashed crops or contaminated water. Humans can also be infected by cats, but if you have a house cat, I wouldn't worry because it's really not that common, because how often do you really go sticking your face in the litter box, right? But as Jamie said, an estimate of 30 to 50% of the world population is actually infected by T. gondii, and that's because there is no known cure. Once it is in your system, it can exist dormantly in cells for your entire lifespan. You cannot cure it, your immune system can't detect it, and it just sits there in cysts in your brain tissue and your muscle tissue. Now, it's more commonly in low-income areas like South America and Africa, but about 12% of the US population is infected, which means more than likely at least one person in this room has latent toxoplasmosis, so you probably had this at some point in your life. Now, this map is a little misleading, because it doesn't show a lot of Africa. Now, this map of infected pregnant women gives a better estimate of the different range of T. gondii. Because as you can see, especially in sub-Saharan and northern Africa, Indonesia, and South America, it is an extremely prevalent parasite. And it's very important to look at the effect of T. gondii on pregnant women, because they're significantly more effective, because it can actually pass from the pregnant woman to the fetus, causing a number of birth defects, from early delivery to miscarriage to blindness and mental disability after birth. Now, it's not incredibly common because you have to have active toxoplasmosis for this, which is you have to be infected 3 months before pregnancy to throughout pregnancy, but it can.
Sorry, is the room reserved for 7, or are you going to be done soon?
We have it reserved from 6 to 8.
We have reserved for a while longer, yeah.
Okay, so there may have been a mix-up. Let me talk to my advisor. I thought we had 7, 9, 7. Okay.
After what you've all been waiting for, organic chemistry. Or as our esteemed Dr. Bradley Hammer would say, the how of it all. So it all relies on one essential amino acid called tryptophan. Now I'm just going to walk you through this little diagram over here because it looks a little confusing at first. There are so tryptophan is an essential amino acid for both humans and T. gondii, and it can be derived to a number of different chemicals down these 3 different pathways. We're going to focus mostly on these 2 on the left here. So when exposed to TPH, which is just an enzyme that causes it to go down the serotonin pathway, it can be formed to the neurotransmitter serotonin and melatonin. I'm sure you've heard of those. Serotonin is necessary for a number of different biological processes, and melatonin is useful for your sleep cycle. On the left here, tryptophan can also be turned into chironine and chironuric acid, which don't have a whole lot of uses, but it's important because that means the T. gondii can't use it. Now, T. gondii seriously relies on melatonin for its metabolism. So what it does is it goes into your cell, it takes the tryptophan, and it forces it to go down this serotonin metabolic pathway so it can eat it. But the issue is that your immune system clearly wants to get rid of the T. gondii, and it understands that it needs to do this. So what it does instead is it releases a huge amount of this TDO enzyme, which forces all of the tryptophan reserves in your system to convert down the left kyranine pathway and eventually to kyraninic acid. Now this has a 2-fold approach. Firstly, it just starves the T. gondii because it can't eat the tryptophan anymore. And secondly, it actually forces the apoptosis in the starvation of T. gondii cells. Basically, it kills the cells that the T. gondii are in. So it kills the cells and they can't replicate anymore. So what we end up getting here is actually just a race of metabolites. The T. gondii is racing to use up all your tryptophan stores and turn it into serotonin, whereas your immune system is racing to take all of your tryptophan stores and turn it into kyraninic acid. Which it causes is a depletion of tryptophan in your system and a serious buildup of serotonin and kyraninic acid, which will be important later. So now for the real important stuff, mind alterations in humans. Firstly, let's discuss mood swings. We already established, as Jamie said earlier, that there can be a lot of effects of T. gondii on the moods of humans and in rats. A study from Kakero et al. In 2016 looked at the effects of latent T. gondii infection on aggression. As you can see here with the light gray bars, those who were infected in the case study with T. gondii showed significantly higher rates of aggression versus the control group in black. Furthermore, suicide attempts in particular were a very strong correlation. So a lot of that aggression came from suicide attempts. This may very well be due to unbalanced neurotransmitter levels, especially dopamine and glutamine. The next and more interesting one is car accidents. So it's been well known for a while that T. gondii infection can actually reduce your reaction time pretty severely. Flegger et al. Looked into this in 2002 and found that subjects with latent toxoplasmosis have significantly increased risk of traffic accidents over time. And first, I'd like to see this. If you don't know anything about statistics, a p-value of 0.0001 is insane. This is an unmistakable correlation from infection to the odds of you getting into a car accident. And this here is actually the antibody titer. What this means is that as the amount of antibodies in your system increases, you are more likely to get into an accident. Which means as time of you getting in the infection goes on, your risks are lower. But what's most important is that even people that had been infected years prior still showed a statistically higher risk of getting into a car accident than those who had never been infected, suggesting that T. gondii can actually affect your reaction times for years after infection. And lastly is schizophrenia. Firstly, the dopamine hypothesis of schizophrenia, which has been a theory that's been around since the 80s, which basically says that schizophrenia is at least partially caused or correlated with basically a screw up of the dopamine receptors in your brain. The dysregulation both up and down, so you either take up too much dopamine or you don't take up enough. It's highly correlated. Laurel et al. In 1996 looked into this, and I actually want to talk about this study for a second because this is wild. This just proves that you could do anything you wanted in the 90s. What they did was they took about 200 people. They injected them with, IBCM, which is just a radioactive chemical that binds to the D2 dopamine receptors. And then they took the baseline, said how active are the receptors, and then they basically just injected all of them with meth. They took an amphetamine that would spike their dopamine levels and just gave it to all of them and saw what the levels would be, or how active the dopamine receptors would be on the schizophrenic people versus the normal people. It's crazy. You could not do this anymore. Like we said, you can experiment on rats. Apparently, you can do it on people. And what we see here is, again, a very small p-value, which suggests, again, statistical significance, and a very clear correlation in the rate, or in the difference of dopamine take-up by the schizophrenics than the baseline. Now, what that means is that when injected with the amphetamine, those who had schizophrenia had a hugely increased rate of dopamine take-up versus the baseline, which suggests that there's definitely something going on with dopamine and high levels of dopamine in schizophrenia patients. And the increased local concentration of dopamine can cause this dysregulation of dopamine receptors, suggesting that if you have high levels of dopamine over time, it can actually cause this dysregulation, which can actually be caused by T. gondii, as I mentioned previously. But wait, there's more. Because I bet all of you forgot about the second metabolite I talked about with tryptophan, hyaluronic acid, which, as Muller and Schwartz mentioned in 2007, hyaluronic acid is a potent antagonist of N-methyl-D-aspartate receptors, blocking NMDA receptors, causing schizophrenia-like symptoms or hypofunction of glutamate neurotransmission. Which, put more simply, is just an increase in TDO activity and accumulation of hyaluronic acid in the CNS. The point is, when you have a lot of hyaluronic acid, it causes the NMDA receptors, which is just a common receptor in your brain that's affected by dopamine and glutamate, which I mentioned earlier, to stop working. If there's hyaluronic acid in the area, the receptors just stop working. And when you block this NMDA receptor, what we find is that what happens looks a lot like schizophrenia. And then when you have the additional activity caused by T. gondii, it can cause symptoms very similar to schizophrenia because of this buildup of hyaluronic acid. So we have actually a twofold issue. When we have a lot of dopamine going on, it can cause symptoms related to schizophrenia, and when you have a lot of hyaluronic acid in the area, it can cause symptoms like schizophrenia, and T. gondii produces both. So in conclusion, T. gondii exists latently in 30 to 50% of all people. Once you have it, you will have it forever. Both of the compounds that build up in T. gondii are correlated very highly with symptoms of schizophrenia, and it has the potential to cause schizophrenia-like symptoms in these infected individuals. It has the unique ability to bypass the blood-brain barrier and directly form cysts in your brain that can change neurotransmitter levels for God knows how long. And the T. gondii can affect behavior and reaction times for years after infection. So what does this all mean? Is there ever going to be some fungus that turns us into zombies and causes us to, I don't know, eat each other? Probably not. But what we do end up having is these microscopic little guys getting into your brain, forming cysts, and changing your behavior for years to come. And honestly, that's a little bit more scary to me. Thank you all.
Hey, sorry, we got confirmation from
Okay.
our advisor